A very good evening to everyone present here today. I, Shreya Saraya, am a genetic counselor by profession and a rare disease advocate for Organization of Rare Diseases India. I thank ORDI for letting me an opportunity to moderate today's Care for Rare webinar. Care for Rare webinar series aims to be a knowledge resource sharing platform to support our common goals of advocacy, awareness, and education of rare diseases with emphasis on its current scenario in India. Today we have with us Dr. Sheth to give a talk on Lynch Syndrome, Diagnostic Approaches and Prevention with Aspirin in Hereditary Cancer. Dr. Harsh is an assistant professor and head of Advanced Genomic Technologies Division at Fridge Institute of Human Genetics, Ahmedabad. He obtained his PhD in Translational Genetics from Newcastle University, UK, under the guideship of Professor Sir John Byrne in 2015. He has diverse experience in areas of hereditary cancer detection and prevention, personalized medicine, statistics, and next-generation sequencing. To date, Dr. Sheth has published 17 peer-reviewed papers, one book chapter, and is co-inventor on an international patent. He is currently leading one of India's largest research work on Lynch syndrome detection and prevalence. We welcome you, sir. Before we begin, I would request everyone other than the speaker to kindly mute themselves and switch off their videos. Participants can post their questions in the chat box, which will be attended at the end of the talk. I now request Dr. Sheth to kindly take over.
Thank you, Shreya. Thank you, Prasannaji, for inviting me here to give this talk. And good evening, everyone. And I see there is one person from Ireland. So good afternoon, where you are. Let me just put up my, so I presume everybody can see my screen?
Yes, sir. It's visible.
Excellent. So Shreya, thank you once again for the one introduction. That saves me a bit of time. I can spend a little bit more time giving this talk. I started my life doing my undergraduate work and PhD work at Newcastle University, and majority of my time was spent working on hereditary cancer detection and prevention. I initially worked on medulloblastoma, and then started focusing on hereditary bowel cancer with Sir John Byrne. So before I give this talk, I briefly wanted to provide some details regarding conflict of interest that may be perceived during this talk. The first one is that I, along with Faye Elliott and Professor Tim Bishop at Leeds, carried out a randomized controlled trial, which is one of the world's largest trial on cancer prevention, Lynch syndrome with aspirin. I'm also a co-inventor on an international patent describing a sequencing-based MSI assay in colorectal cancer, about which I'll be talking today. Investigator on a study, an ongoing study that is currently looking systematically at the prevalence of MSI and Lynch syndrome prevalence in colorectal cancer patients in India. I'll also be providing some brief data on that. I'm also co-investigator on studies investigating utility of sequencing-based MSI assay in cancer samples across India, Malaysia, and Indonesia. This work is done in tandem with Newcastle University Malaysia campus and University of Gajah Mada in Indonesia. My current research work is funded by Gujarat State Biotech Mission, and a small part of my work is also funded by UPC Research Council. And lastly, I'm a panel member of Scientific and Technical Advisory Panel of South Africa's Open Genome Project, which is working towards implementing genomic technologies in routine healthcare across South Africa. So like I said, I spent around 10 to 11 years in the UK going off an undergrad student to being a PhD student and then becoming postdoc. And it was just about 3/4 of years ago that I shifted my base from Newcastle to this lovely city, Ahmedabad, where I also grew up. And I placed my base at Fridge's Institute of Human Genetics. Now, during these 13 years that I've been conducting research, I've had a breadth of experience, starting with the pharmacogenetics of warfarin, where I worked with Professor Ann Daly and Farhad Kamali. They are the legends when it comes to the pharmacogenetics world. I was also involved in developing rapid molecular diagnostics, along with then spin-out company called Quantum DX, which is based in Newcastle. During my PhD and postdoc, I was also involved in de novo mutations in severe male infertility phenotypes, and also looking at the role of aspirin in colorectal cancer prevention and hereditary cancer detection, especially Lynch syndrome. In the past 1.5 years, I have started to focus more towards sequencing-based MSI testing in India, developing various different genomic assays for diagnostic purposes, but also looking at genomic landscape of autism spectrum disorders. But today, I want to focus solely on hereditary cancer detection and prevention. And the sole impetus behind this work comes from this one line that was written by Thomas Adams in 1600s, where he says that he's a better physician that keeps diseases off us than he that cures them from being on us. Prevention is so much better than healing because it saves the labor of being sick. And I'm sure that not only doctors, but the patients would agree that it's better not to have cancer if you can work towards it. Now, the world of genetics in oncology became very, very famous because of one person, that is Angelina Jolie. In 2013, when she released a public statement stating that she had carried out double mastectomy because she was at high risk of having breast cancer, she had one of the copies of her BRCA1 gene had a pathogenic mutation in it. So in order to prevent breast cancer, she had done double mastectomy. And because of that, in the year 2014 in the UK, there were the referrals in breast cancer clinics nearly doubled and has been at that level ever since. And she essentially set the trend of not only doing identification of risk, but also ameliorating it if possible. So this is where the whole world of genetics comes to play in the field of oncology. If you can prevent it, that saves you the trouble of getting sick with cancer in the first place. Now, when she talked about having one of her copies of BRCA1 gene affected, and because of that, her risk of cancer increasing, there was one thing that escaped everybody's attention. That is Knudson two-hit hypothesis. Why is it that if just one copy of the DNA is affected, does your risk of cancer increases? And to understand that, you have to go back to 1970s when Alfred Knudson saw that patients where one of the copies of retinoblastoma gene had a mutation, those patients ended up with retinoblastoma at an earlier stage compared to patients where both copies of the genes were intact. And that essentially led to the idea that at least you need one copy to be affected to have a risk of cancer increase dramatically, because of which you would have cancer at an earlier age. To put it in simple words, say if you have two bodyguards just outside a hotel, and if one of them goes to the loo, you at least have one more person standing there guarding the place. So if something goes wrong, you can still look after it. So to put it in the world, or put it in the context of cancer, you still have at least one intact copy. You have two intact copies. So if at least one goes wrong, you still have one to look after you. But if you just have one bodyguard standing outside your hotel and he wants to go to the loo, well, then you have nobody looking after the place. And because of that, you are at a higher risk of having something going wrong. Or to put it in an analogy of cancer, you are at a high lifetime risk of getting cancer. And this entire hypothesis can also be seen in colorectal. So to briefly tell everyone, colorectal cancer is one of the most common cancers you see in India. You have over 30 to 40,000 patients that are affected with it every year. Now that number may sound low in context of oral cancer, but these cancers can be very aggressive from time to time. Now, generally, when the natural progression of cancer generally goes from a normal-looking epithelium at one end, so that's how your gut actually looks like. And once you end up accumulating a mutation in one of the genes, you see a small bulb coming through. Now, that's a small adenoma, which then turns into a massive polyp, which then turns into a full-blown cancer, and that goes through metastasis. Now, on an average, 5 out of 6 patients that oncologists see in their clinic end up having these cancers because of the FAP pathway, or what is known as familial adenomatous polyposis pathway. This is where you have hundreds to thousands of little polyps across the line of your gut. And that happens because you have a mutation in the ABC gene. In about 1 in 6 cancers, however, you see mutation coming up in one of the mismatched repair genes. So you would have MLH1, MSH2, etc. And because of that, you don't have hundreds to thousands of polyps. You instead have something known as sessile serrated adenoma. So these are very small polyps that can barely be seen, but they have a good potential to become cancer down the line. And this pathway happens in 1 out of every 6 patients that an oncologist would see in the clinic. And it's these patients that are at high risk of having hereditary cancer, also known as Lynch syndrome. Now, Lynch syndrome may be a connotation that is being used today, but scan back 10 or 20 years ago, it had a rather complicated name. At that time, it was known as hereditary non-polyposis colorectal cancer, simply because you didn't have hundreds of thousands of polyps in your bowel. And this whole work came because of this one very famous family known as family G, where multiple members had cancers across the GI system. Or in some cases, they also had cancer in the endometrial or in the womb. And at that time, Dr. Henry Lynch worked heavily towards it to try and understand not only the epidemiology, but the genetics aspect of it. And it was in his honor that this disease was later named as Lynch syndrome. Now, it was in just 1993, that's 27 years ago, that the very first gene was identified for this. It's known as MSH2. And subsequently, 4 other genes have also been identified that causes Lynch syndrome. So Lynch syndrome is an autosomal dominant condition. You just need 1 copy of your DNA to be affected to then increase your risk of getting cancer. And that happens when at least one of the copies of the 5 mismatch repair genes, well, I've written 4 here, but there's a 5th one now. It's MSH2, MSH6, MLH1, PMS2, and now there is EpCAM as well. So if any 1 copy of any of those genes are affected, you are at high risk of getting cancer. And mostly you would have cancers of colorectum, that's your gut, or your womb, or upper GI or genitourinary cancers. The striking thing is that early last year, World Health Organization estimated that at least 1 in 125 people worldwide are Lynch syndrome carriers. And that is something very striking because these patients do have high lifetime risk of getting cancer. To give you an example of how high that is, here are the data that's been taken from prospective Lynch syndrome database. So based on which gene is affected, your risk of having cancer increases across your lifetime. And as you can see, your risk of colorectal or endometrial cancer can shoot up to 50 to 60% throughout your lifespan, which is extremely high. And that is why it's extremely important to identify these individuals and, if possible, try and prevent cancers from occurring in the first place. Now, currently, if you want to identify these patients, what you have to do is you would have to wait for 1 patient to end up having colorectal cancer, in which you would carry out a microsatellite instability testing or immunohistochemistry of the 4 mismatch repair proteins. Sorry, just a moment, please. If your microsatellite analysis comes stable or all four proteins are intact, well, it was a sporadic cancer. But if you have microsatellite instability or one of the four mismatch repair proteins are absent, well, then you then have to do BRAF or a BRAF E600E or MLH1 hypermethylation testing, because that would then help you to stratify whether the patient is likely to be Lynch syndrome carrier or not. If you have the mutation or you have a hypermethylated MLH1 gene, well, again, it was a sporadic cancer. But if you don't have the V600E mutation or you don't have hypermethylation, well, now you would have to do germline testing for the 5 mismatch repair genes and then try and identify the mutation that would help you to confirm the diagnosis of Lynch syndrome. So a fairly complicated pathway to try and identify a person with Lynch syndrome. Now, I use the term microsatellite instability or microsatellites, and I know many of you may already know about what this test is. But for people who don't, well, in your DNA, there are places where the letter of the DNA keeps on repeating itself many times, like I've shown here, and these are known as microsatellites. Now, what happens is if one of your mismatch repair proteins is not working very well, you end up accumulating errors in these regions of the DNA, because essentially your mismatch repair proteins are like spell checkers, kind of like your Word document. If you misspell something, the Word document autocorrects that word. In the same sense, your mismatch repair proteins comes along, tries and identify an error that has occurred during copying process of the DNA, and repairs it. But if your mismatch repair system is broken down, well, these errors would keep on accumulating, and these errors are known as microsatellite instability. Now, because of its importance, UK's National Institute of Health and Care Excellence and US's FDA now recommends MSI testing across all colorectal cancers to identify Lynch syndrome patients. The problem, however, is, like I said, one in 6 patients that come in your clinic are likely to have microsatellite instability. And currently, there are 2 ways to detect it. The first one is immunohistochemistry. Now that's a bit more old school. You need experts to look down the microscope and see whether a protein is present or absent. The second method is known as fragment length analysis. Here, you use a PCR instrument and then take it down a fragment length analyzer to see to assess the length of those microsatellite regions. Now, again, this is a slightly advanced system, but you need manual interpretation. And because of that, sometimes you get inter-operator discordance. So the results can vary if one person is looking compared to the other. They're not amenable to high throughput because somebody has to be there to see the results. And because of that, cost per sample is not affordable in low income or low resource settings, especially in a country like India, where these tests cost 6,000 to 10,000 rupees. That is excruciatingly high. And because of that, it is estimated that worldwide, 3 to 4 out of 1,000 people in the population carry mismatch repair gene defects, but only 10% of those are known to medical services. So that is actually very, very poor rate of diagnosis. And we're missing out on patients who could actually receive help in reducing the risk of cancer. So clearly, there is a need for a high throughput, automatable, cost-effective MSI assay. More like a Gatling gun. You essentially want to have a huge number of samples that you can then analyze very, very quickly. Now, this is where, along with my 2 colleagues, Lisa Redford, who is now working in Norway, and Dr. Richard Gallen, who is currently senior postdoc in Professor Sir John Burns' group. Together, we developed this assay. It's a sequencing-based MSI assay. Now, out here, I would also like to point one more thing, that currently there are several different commercial entities that offer sequencing-based MSI assay. The problem, however, is they are extremely costly, because not only are they looking at microsatellite instability, but they are testing hundreds of different genes simultaneously, or they have huge number of microsatellite markers that they're looking at concurrently, which makes them very, very expensive to run. Our approach was slightly different. We were looking at microsatellite regions that were only 7 to 12 letters long. That means they are sequenceable and with slightly less error rate. More importantly, every microsatellite that we chose was sitting right next to a polymorphism. And I'll tell you why that was the case. Now, our assay did two things to call microsatellite instability. The first thing is to see how many letters were added or deleted in tumor's DNA. As you can see here, compared to the matched normal, you have a much higher rate of a single letter or two letters missing in those microsatellite tracks. The second thing that our assay did was to look at allelic imbalance. That is to say, if your microsatellite is sitting right next to a polymorphic variant, especially if it's heterozygous, then what happens is, if you have an error that is induced by a PCR or a sequencing machine, that error would be shown across both alleles. But if it is a genuine microsatellite instability mutation, then it should only occur on one allele or one copy of your DNA. And as you can see here, very, very clearly, you do see that allelic imbalance coming through, especially in this lane. You can very well see that this is a microsatellite unstable tumor cell line, and you see this deletion event occurring on just one allele and not other. So it's a genuine event. So using these two approaches, we developed a naive Bayesian model to carry out automated MSI calling. And we used 24 markers to carry out MSI testing. As you can see on your right, we first took around 100 samples to develop a classification system. So the samples that are highlighted in red are MSI-high. The samples that are highlighted in blue are MSS, or microsatellite stable. Now, just to see if there was redundancy in these marker panels, we did manage to prove that just by looking at 6 markers, we could still call MSI accurately. Not only that, when we took a validation group of 100 samples, we managed to achieve 100% sensitivity and specificity, which is excellent. And that was not just with the 24 marker panel, but also with the 6 marker panel. On top of that, like I had shown previously, we still need to do BRAF testing in these patients to differentiate whether a patient is likely to be Lynch or sporadic. So we incorporated BRAF V600E mutation testing within the panel in itself, along with KRAS codon 12 and 13 mutation. These are they tend to mutate very, very frequently in these patients. So these markers were added at no additional cost. And as a result of that, our sample workflow was very, very simple. You simply capture these 24 markers, you amplify them, you prepare, and you sequence on an Illumina MiSeq platform. Not only that, once you have the data, you simply put it on a computer, run the algorithm, and you get automated telling of whether a sample is MSI or MSS. Now, since we have added polymorphisms, single nucleotide polymorphisms within the panel, we are also able to provide traceability so that in case 2 samples are mixed up, we can discard those samples from our analysis. Now, this work was published in Human Mutation last year, and there are now 2 worldwide patents on it. One is already published, one is patent pending. And I'm a co-inventor on the second patent. Now, as some of you may already know, MSI testing is very, very important, not only because it helps you to diagnose Lynch syndrome patient, but it also helps you to provide prognosis to patients. On top of that, nowadays, it's crucial to do MSI testing before you give immunotherapies, because if you have an MSI-high cancer, then you're more likely to receive benefit from taking immunotherapies compared to if you have an MSS cancer. Not only that, if you're able to identify a person as having Lynch syndrome, then it has implications on surgery as well. You might want to carry out panproctocolectomy rather than removing just a small part of your bowel in order to risk of cancers coming in the future. And lastly, if you have identified a patient as having Lynch syndrome, then you can start them on prophylactic aspirin chemoprevention treatment, or you can then start them on regular colonoscopies. Now, this work so far was done at international scale. When we started to look at India, there was a major problem. The incidence or prevalence of MSI and Lynch syndrome in cancer patients in India is still unclear to date. And there are several reasons for that. Many studies have been conducted, but either they were done in very small sample cohorts, or they were just looking at one thing or the other, but they have never looked together systematically. So my group received funding from Gujarat State Biotech Mission to study MSI and Lynch syndrome prevalence in colorectal cancer patients in India. Our aim is to analyze 300 cancer patients till 2022. Within the study, we'll be carrying out MSI testing, BRAF testing, as well as germline mismatch repair gene testing in these patients. It's a first of its kind study in India, and it's extremely critical for preparing care pathways and cancer prevention strategies, because if you don't know how common or rare a disease is, how can you prepare a healthcare pathway? Now, this is an ongoing study. Our recruitment will be open till the end of 2021. We are more than happy for anybody to collaborate with us. Currently, our collaborators are dotted across India. So we have most of our collaborators are situated in Ahmedabad. A couple of them are situated in Mumbai. Now, what I'm going to present to you is preliminary data that was analyzed till March 2020. Following lockdown, recruitment is still going on. But since the numbers are still being analyzed, it would have been premature to show the results. Till 31st of March, we had recruited overall 55 patients. The mean age of cancer diagnosis was 51 years. So that's in concordance with what has been observed in the literature in India. So that's a fairly young age to look at. Not only that, around a third of the patients had cancer in the ascending colon. So that's the right-sided part of your large intestine. And that is where you see a high prevalence of MSI-high cancers. So when we did MSI testing in these, about a quarter of these patients had MSI-high cancers. So that's a slightly higher rate compared to what we see in the West. In the West, we see 1 in every 6 patients. In our cohort, we are seeing 1 in every 4 cancers. 1 in every 4 patients, my apologies. And after doing BRAF testing, when we did germline mismatch repair gene testing, we found that majority of the mutations were creeping up in the MLH1 gene. Well, subsequent to that, we have seen a couple of mutations coming up in PMS2 gene and MSH2 gene as well. But predominantly, mutations do creep up in MLH1. We did have 2 patients whereby they had microsatellite stable tumors, but they had a very strong family history of cancer. So we did germline testing in these patients, and lo and behold, we found a pathogenic mutation in the MLH1 gene. And in the second patient, we found mutation in the CDH1 gene. Now, to briefly conclude this study, this is the first assessment of MSI-high phenotype and Lynch syndrome prevalence in India. And we do see a slightly higher rate of MSI-high and Lynch syndrome prevalence in our patient cohort compared to the European population. But I would be cautious to say it out loud at the moment, because our patient pool is small and it is attuned to some form of recruitment bias. But we'll have firm data to you by 2022. So far, majority of the patients have mutation in the MLH1 gene. And like I said, the recruitment is ongoing. So if anybody would like to collaborate, please get in touch with me. Now, the problem is, if you have diagnosed somebody with Lynch syndrome and their family members as well, you know this. And if they are at high risk of cancer, you would like to help them out to try and reduce that risk. Sorry. So back in 1980s, a whole series of cardiovascular observational studies were out, whereby people observed that patients who were getting aspirin had lower risk of colorectal cancer compared to patients who were not taking aspirin. So that naturally led us to think that what if we were to give aspirin to patients who have Lynch syndrome, right? But they haven't had cancer yet. What if we could try and reduce the risk of colorectal cancer in those patients? So CAP2 trial was started back in the 1990s. And the idea was you would recruit 1,000 patients with known Lynch syndrome, mutation. You would then give half of these patients 600 milligram aspirin every day, and half of these patients a placebo. And it is a double-blind randomized control trial. You'd give them aspirin for 2 years, and then you would follow them up for 10 years. Late last year, we managed to complete a full follow-up up till 10 years. And not only that, in some patients, because in the UK and in Finland, these patients were part of a cancer registry. So for some of these patients, we even had data up till 20 years. So we conducted analysis of this trial. So I, along with Faye Elliott and Professor Tim Bishop at Leeds, were involved in the analysis of this work. And this entire trial was led by Professor Sir John Byrne, who was my supervisor during the PhD. And the results of these trials were published in Lancet just a few months ago. Now, like I said, we had data for 10-year follow-up on 861 patients, and we had 20-year follow-up data on 340 out of those 861 patients. Our primary endpoint was to see reduction in risk of colorectal cancers. And briefly, what we observed was that patients who were given aspirin had only 40 colorectal cancers, whereas patients who were given placebo had ended up with 58 patients had colorectal cancer. So there was a reduction of 18 colorectal cancers overall. Now, there were two ways to look at the analysis. The first one is known as intention to treat. That looks more like a real world scenario, whereby even if you have randomized patients to either aspirin or placebo, they may not take these interventions all the way up to 2 years, right? And you want to see whether you observe a risk reduction in cancer in these patients. Lo and behold, we did observe, and there was around 35 to 42% risk reduction in these patients. Now, again, there were two ways to look at the data. The first way was to see whether you were able to reduce time to the first, or you were able to reduce risk of the first colorectal cancer. So that is encompassed by a value known as hazard ratio. And that is where we saw 35% risk reduction. The second way to look at is incidence rate ratio. In Lynch syndrome patient, it's not like you just get one cancer and it ends. They are at lifetime risk of getting multiple cancers. So you want to capture all of that data, and incidence rate ratio does that for you. And this is where we saw 42% risk reduction in colorectal cancer. However, what was interesting was to look at poor protocol analysis. So these were the patients who took aspirin religiously for 2 years or more. And in those patients, we saw a 50% reduction in risk of cancers. So to summarize, 600 milligram aspirin a day taken for more than 2 years reduces risk of colorectal cancers by 50%. And remember, this reduction was observed all the way up till 20 years after they had stopped taking aspirin, right? And looking at this, what we can say is that if you have 24 Lynch syndrome patients, and if you were to start giving them aspirin, you would at least prevent one colorectal cancer from occurring. And that's a massive number. That's a very, very powerful number. The second analysis that we wanted to do was to look at all Lynch syndrome cancers. So not just colorectal cancer, but also look at endometrial cancers, upper GI cancers, etc. Now, again, we managed to prevent 15 cancers in the aspirin group compared to placebo. But when we looked at intention to treat analysis, we didn't see a statistically significant difference between aspirin and placebo group. But there was definitely a trend in risk reduction. However, when we looked at the per protocol analysis, here we saw a sharp 35% reduction in risk of all Lynch syndrome cancers. The majority of that risk reduction signal was coming from patients with endometrial cancers. That's where we saw the biggest benefit. The problem, however, is that it was not, strictly speaking, statistically significant. So the trialists wouldn't like it, but there was definitely a significant trend. Now, this is just a this is a randomized controlled trial. Beyond this, there have been several observational studies, several randomized controlled trials, all suggesting and giving evidence that aspirin does reduce risk of colorectal cancer in Lynch syndrome patients. Now, heeding on to these data, UK's National Institute of Health and Care Excellence earlier published a guideline stating that patients who are identified as having Lynch syndrome should be prescribed aspirin daily for 2 years or more. The problem, however, is 600 milligrams is actually quite a high dose to be given to patients. So we want to know what is the optimal dose that could work. Could 100 milligrams a day work, 300 milligrams, or 600 milligrams? So CAP3's trial started a few years ago, whereby patients were stratified to 3 different groups, where in each group you are given 100, 300, or 600 milligrams aspirin daily. And these treatments will be given for 2 years and will then be open label. So the patients will be able to know what dose they are taking. And then they'll be followed up for 3 to 5 years. Recruitment overall, 1,900 patients to date have been recruited. And the first set of data will arrive in 2025. The problem is that's very, very long to wait to identify which dose to give. So we can use some data that is already available from other trials, such as Aspirin or Arrive, Seafood, or Aspect. And in those trials, what we observe is that low-dose aspirin don't reduce cardiovascular or indeed cancer events in big people. When I say big people, means these are people who have got high BMI or high body surface area. So in those patients, giving slightly higher dose might be beneficial. However, if these patients are old or frail with specific risk factors, then they are at extremely high risk of having GI bleeds or much more dangerous hemorrhagic strokes. Right now, these strokes occur 1 in every 10,000 people in the general population. But as you grow older, this risk increases substantially. So there was one more observation that was also made, that in the first few years that patients were given aspirin, there was a slightly higher rate of cancer and deaths. It's still very, very difficult to explain why that is, but it is evident. So these are the 3 things one could think of. If you have a high BMI or body surface area, then you might want to have more than 100 milligrams a day of aspirin to achieve the desirable effect. Aspirin overall may be destroying early neoplasms or reducing metastasis to provide its anti-cancer effect. But earlier in its track, it can promote lesions that are fairly advanced. And that is why you might see an early rise in cancer rate, followed by a dramatic reduction in cancer rates. People who are old and frail should not be recommended aspirin. However, if you are less than 50 years old or or you are just about to retire, that is a decade before retirement, then you might want to think about taking aspirin if you are identified to be a Lynch syndrome carrier. Of course, you should be talking with a healthcare practitioner before you start any self-medication. In conclusion, what I have demonstrated is that Lynch syndrome is more common than expected in the general population. Data on Indian population is currently being analyzed and will be available by 2022. Detection of Lynch syndrome patients can now be done with high throughput technologies. We have developed one of those technologies as well and is available right now. We can now prevent cancers in Lynch syndrome patients, not only just colorectal cancer. Just 2 hours ago, UK's National Institute of Health and Care Excellence also recommended doing MSI testing in endometrial cancer patients to identify Lynch syndrome individuals, because then you can provide these chemopreventive interventions in those patients as well. Based on all the data that we have on aspirin so far, UK's as well as recommends giving low-dose aspirin to individuals who are identified as Lynch syndrome carriers in order to prevent cancers later down the life. The optimal dose right now of aspirin is still up for debate, but the first set of data will arrive in 2025 as part of the CAP3 trial. In terms of optimal duration, right now, looking at CAP2, two years look reasonable. But there is some data coming from observational studies that shows that if you take aspirin longer than 2 years duration, you might see improved chemopreventive effect. But more data is required towards that. In the end, for all of this work, obviously, it's not a single person work. It's done by a fantastic team here, not only at the Institute in Ahmedabad, but throughout the world, led mostly by our international as well as national collaborators, our wonderful funders, especially Gujarat State Biotech Mission, Cancer Research UK, and Medical Research Council. And with that, thank you all for listening.
Thank you very much, Dr. Harsh, for a comprehensive and an enlightening presentation. So, shall we take the questions now? Yeah. So we have a question from an ORDI member. How cost-effective is the MMR protein test? Should all Lynch syndrome suspects get tested for MMR?
Okay. So right now, at least across the world, it is recommended that if you have colorectal cancer, you should get tested for microsatellite instability or MMR testing on your tumor. If you are, and that has been shown to be cost effective. There was a study done all the way back in 2015 by National Institute of Health Excellence and Research in the UK that showed that actually routine MSI testing in colorectal cancer is cost effective. And now that guideline is being extended to endometrial cancer as well.
Okay. I hope that question is answered. The second question is, do you recommend direct-to-consumer genetic testing for individuals at risk?
How do they define individuals at risk? The question is, you do have, the problem with Lynch syndrome is, you can have cases whereby patients don't have any family history whatsoever. So those patients do tend to be left out of net. So right now, the approach that is being utilized is if you have cancer, you should get tested for MSI. And then if you're MSI high, do BRAF or MLH1 testing and then do germline testing. If you are identified with germline pathogenic mutation, then your first degree relatives are likely to be risk at Lynch syndrome as well. In that case, yes, you should get tested for that mutation in first degree relatives.
Thank you. And we have one last question. Are women with Lynch syndrome more likely to develop breast cancer? If yes, what is the molecular correlation?
Okay. That's slightly tricky. But yes, in Lynch syndrome, you do find some women with risk of breast cancer, but it's extremely low compared to if you have mutation in the BRCA1 gene. The problem is BRCA1 gene, along with your BRCA1 or your mismatch repair genes, are all involved in repairing your DNA. So if you do see a breakdown in that mechanism, you do end up with mutations occurring all over the place. And if that occurs in your breast cancer, in your breast cells, then yes, it could lead to breast cancer, but the risk of breast cancer does tend to be relatively low. The highest risk actually would be of endometrial cancer in women.
I hope the questions are answered. Thank you so much, sir. And before closing the session, I once again thank Dr. Harsh for his wonderful talk. I thank all the participants whose presence ensure the success of today's session. And lastly, I would like to thank the sponsors and the ORDI team. Thank you very much.
Thank you so much. Thank you all for listening.